And if I could have the house lights, thanks very much. If I could have the house lights up, that would go fully up as maximum voltage. That would be great. So my poor eyes, the aging eyes, the thickening lens, that's fantastic. It's almost daylight. I will thank our sponsors in a moment. It's time for questions. If you can raise your hands, we have 4 people with microphones who'll come to you and hold up their hands and frantically wave and hopefully gain our attention. It's very kind of you, Linda, to agree to be questioned and take the calls. When I was listening to you talking, I thought, this is fantastic. All roads lead to this receptor hormone interaction. Do they really? I mean, it sounds too good to be true that it's as simple as that.
I think it probably is as simple as that as far as that system goes. It really seems to work in the animal models. It's beginning to look as though it will work in people, although of course we don't know to what extent. I think there's probably also a lot that we're missing. I mean, what happened was somebody asked a worm what would make it live a long time, and then we asked whether the same things would happen in other animals and humans. Almost certainly there are things in humans that we don't yet know about and that are going to be very important.
What do you suspect from what you know of humans to date? I mean, certainly these growth factors, these insulin-like growth factors are heavily implicated in cancer, seem to be the mediator for obesity and cancer. But that's, again, disease rather than aging.
Indeed. I think the whole axis of inflammation, so overactivity of the immune system during aging, that's very much a hallmark of aging in people, very easily sampled by looking at inflammatory markers in the blood. I suspect that's going to be very important in people, and it's not very easily modelled in these little invertebrates that I was talking about.
What about mitochondria in all this? Where do they fit in? Because there's a hot sort of area of research in mitochondria, those little energy boxes inside our cells.
There is indeed. So, I mean, those of you who are at all familiar with, the biological literature and what goes on in aging, somebody called Harman back in the 1950s proposed that the reason that we age is that we oxidize, basically. Oxygen's bad stuff. It can cause damage to macromolecules. And these little organelles, the mitochondria, they're the powerhouses of energy in the cells, and as a byproduct of their activity of using nutrients and making energy molecules, ATP, they produce what are called reactive oxygen species. They're slightly modified versions of oxygen itself or of things derived from it.
Free radicals.
Free radicals, exactly. And they can damage, proteins, fats, and so on in the body. But, people have done loads of experiments on that recently, trying to directly modify the production of the free radicals or the systems that protect against them in cells. And it seems to be quite easy to make an animal sensitive to oxidative stress if you knock down those defense systems, but it doesn't live any longer. It doesn't look as though they're limiting for aging. So although it's a very obvious and attractive idea that our use of oxygen is what's causing aging, actually it doesn't check out experimentally.
Which fits with the randomized trials of antioxidants, where you die sooner when you're taking your antioxidant supplements.
Indeed.
Damn it. Yes. Yep, you're on. If you wouldn't mind standing up so we can see you, that would be great.
Yeah, you made the comment, Linda, that birds live longer than you'd expect them to, based on their size. And is there an explanation for that? Why do birds have a longer lifespan than you would expect when you look at a comparable sized mammal?
With the exception of budgies, I suppose.
I think that's one of the most important questions in the whole of aging research. So we've succeeded in making these animals live, if we're lucky, twice as long as normal. But there's that huge range of lifespans in nature. And you would think that it would be a very obvious thing to just go and try and figure out what it is that's happening in the long-lived ones that we could then come back and look at experimentally or medically. The problem is that there are so many other differences between different species. So if you look at a whale and a mouse or a bird and a mammal, they differ in all kinds of different ways. So there are genomic differences, very many. differences in what they're like physically, physiologically, behaviourally and so on, very many. How do you figure out which of those genetic differences are the ones that are producing the difference in the rate of ageing, rather than all those other traits?
Or indeed the extrinsic factors, the small matter of seawater.
And all that. So it's really, really hard to pin down those differences, although people have tried. I mean, it's thrown up a few custard pies. So, for instance, this, oxidative damage theory that we were just talking about. That naked mole rat that I showed that lives so long for its size. People said, right, let's go and look at how much oxidative damage this thing's got up. We think the defence systems will be up. It will be really low. It shot through with oxidative damage. Nobody's ever seen levels of oxidation of macromolecules like you get in the naked mole rat. So that's a tough one. Where I think there's a much better chance is where you've got species where the same genome is producing individuals that live for very different lengths of time. I mentioned the honeybee. So what you have there is females that can either become a reproductive if during development they're fed this royal jelly by the workers. But those very same individuals that don't become queens because they're not fed royal jelly, become workers. And the workers can live only for a few weeks, whereas the queen can go for a decade or more. And I think trying to figure out how that very same genetic material can produce those two very different lifespans might be an easier way in. But even there hasn't been much progress made with it yet. So
yes.
I think it's a really interesting question.
Yes.
At the risk of... So I won't make jokes about longevity of the queen and what the progeny should be doing. I've got a question about the role of ageing versus obesity. I mean, in many of the epidemiological data you showed us, you could replace ageing with adiposity. And the same signalling pathway is present in both. I mean, you've got insulin-based signalling in obesity, you've got IGF-based signalling in ageing. How much of this is being too rich?
Again, a very interesting question. I mean, the role of adiposity is proving a very, very difficult one to unravel, because quite often if you have an intervention like dietary restriction would be the obvious one, where you make the animal lean, it lives longer. But the question is that leanness causal in making it live longer? And I think the jury's completely out on that one at the moment. It's not clear. So, for instance, there are genetically obese mice. They're defective in their response to a hormone called leptin, which is produced from the fat, and they keep eating even though they've got a lot of fat, and they get fat. If you dietary restrict those animals, you can make them as long-lived as a control mouse, but they're still much fatter. So it's not clear that the fat per se is the important thing.
What about insulin signalling, though? I mean, there's a synergy between leptin and insulin signalling that occurs, and those leptin-deficient animals are diabetic from the get-go. How much of that is convergent on insulin signalling, do you think?
I think the jury's out on that one at the moment. It's just not clear what the interplay between the two is. It's very clear that some of the long-lived mice are long-lived because they actually have virtuous fat. So there's a system called growth hormone, which is active during development, and as its name suggests, promotes growth, actually through this insulin-like growth factor 1. And a number of the long-lived mouse models are defective in that growth hormone axis. And it looks as though in those animals, part of the reason for their increased longevity is actually that their fat is doing something that's beneficial to the animal. This has come from various surgical fat removal experiments. So fat's very complicated stuff. It's not just fat. It's producing all sorts of little hormones and chemical messengers.
So liposuction could be life-shortening.
Yes,
Thank you.
it could be.
Yes.
If you've got virtuous fat.
If you've got virtuous fat, well, isn't all fat virtuous? Yes.
Ian McCready from RMIT University. I just had a question about what is the best Alzheimer's chemopreventative. I actually work on A-beta in yeast, so I had a very strong interest in your talk tonight. According to my thoughts, I think simvastatin may be the best. So it's the first blockbuster drug lowering cholesterol levels, and there was a study in the US by Ben Willowsen that showed that it lowered the incidence of Parkinson's and Alzheimer's by about 50%. And he lined them up with other cholesterol lowering drugs, and it was the best in the set, and it was lipophilic, getting into the brain, and most of the cholesterol's in the brain. Have you got any comments on cholesterol and Alzheimer's disease?
Well, I mean, one can talk in terms of risk factors for Alzheimer's disease and lowering them. And apart from age, the major risk factor is actually diabetes. In fact, a lot of people think that Alzheimer's might be primarily, at least in its initiation, if not in what happens subsequently, a vascular disease, a disease of the blood system, and that it may come from vascular damage. So in terms of prevention, anything that prevents diabetes is going to be beneficial. But then, of course, the major genetic risk factor is your genotype at the ApoE locus, which then starts to go into this whole story that you're talking about. So again, as a preventative measure, anything that starts to deal with that axis is going to be helpful. But really, in terms of drugs for Alzheimer's, once it started to develop, my own perception of the current situation is that it is very bleak. There are no good drugs.
So we've got to be getting in there much earlier.
Yeah.
By the way, we would welcome questions from our school students, so please don't be afraid to put up your hand. And there is no such thing as a silly question, so please put up your hand. And, sorry, yes, somebody here's been very patient waiting.
Andrew Trelaw from the Australian National Data Service. As I listened to your talk, I was wondering about the relationship between the work you were describing and Elizabeth Blackburn's work on telomerase, which I may have missed it, but I didn't hear you mention it. And it seemed that they're both dealing with questions of ageing and ageing of cells, but from very different directions. Could you comment on the relationship between those two strands of activity?
So we should just explain for those of the audience who are not biological here, we're talking about the bootlaces at the end of, the bootlace ends at the end of chromosomes, which get shorter as we age or as cells multiply, and telomerase keeps them long.
And eventually, if they shorten too much, you can actually start to erode into the chromosomes themselves and damage genes. Obviously, I think that works absolutely fantastic. But as far as aging goes, I think it may be one of those situations which are frustrating for people like me, where the only good study for humans is actually humans. And the reason for saying that is that the invertebrates are generally what's called post-mitotic as adults. There isn't much cell division in the adults, and telomeres only shorten during cell division. So almost certainly telomeres are not an important player in their aging, so they're not going to be a useful model. But you think the mouse would, because it's got normal mammalian telomeres, and they do shorten during aging. The only problem is that they're incredibly long to start with. So during the normal aging process of a mouse, they don't shorten enough to get anywhere near causing the kinds of problems that you can see with human telomeres. So I think that that's a situation where we just don't have a good animal model.
And is there any indication that IGF has any role there?
Not as far as I know, except in as much as it would promote cell proliferation.
Yep. Yes.
Thank you very much. Emilio Bedoa, RMIT. Again, thank you very much for an absolutely fabulous talk. It's absolutely marvellous. I just wonder if you'd like to comment on longevity versus quality of life. It seems to me that the underlying assumption is with longevity, increase in longevity, there's an increase in quality of life. And I wonder what the evidence for that really is.
Well, I would say straight away that what we're interested in is quality of life, not lifespan. I mean, that's what this kind of work is about. But of course, the increase in lifespan is happening anyway. I think there's a very interesting question there about, with the increase in lifespan, is what we're seeing an increase mainly in the dysfunctional period or partially dysfunctional period at the end of life, or is the whole thing just being kind of stretched out in a linear fashion? Or are we seeing what the demographers call compression of morbidity, so a longer lifespan but with less? And the answer is a terribly difficult question to answer, because what you would need is people making exactly the same health measurements over a very long period of time to see what's happened. And of course, people keep altering their criteria for what they're describing. So I think we don't have a really good set of statistics that have been carried out, where the measurements have been carried out in a standard way.
But it seems to be true. I mean, there's no evidence in the Australian data that there's any compression of morbidity. But it seems, as you said in your talk, that the predictors of a longer life expectancy are also the predictors of a longer disability-free life as well. So it's just shifted to the right.
I think the average story is that it's just being generally pulled out in a linear fashion. I mean, some countries report compression of morbidity. Some say it's lasting for longer. If you look at the average, it just looks as though what's happening is that everything's happening more slowly.
Yes, at the back.
Hi, Raiden Richardson from Melbourne High School. We loved your talk amongst the school community, the school people here. Just a quick one on although the feeling amidst the scientific community that I can see here from people in physics and biology and engineering is really positive and optimistic about this interest in decreasing the effects of aging and fighting it. Do you worry, or are you apprehensive about this research being opposed, I guess, in the wider community by people who aren't of a scientific background and who would have to worry about the ethical side of things or other concerns that they have?
Can I ask you a question? So you're worried
Yep.
about the fact that you might get medications produced which might be harmful, or
Yep.
the ethics of people living longer at the expense of others, that sort of thing? Is that
Or
what-
even perhaps people who fear too much or fear or to such an extent that it's a detriment on the research that you've done, and that it hinders upon it on the optimism that there really is for ungrounded fear, that kind of thing.
It's an important question, the public reaction to
I
it.
think it's an absolutely vital question. I mean, research can always potentially be dangerous because you never know what's around the corner. And I mean, obviously the implications just of people living longer without people like me doing anything, let alone what may come along in the way of therapies for aging-related disease, are going to have the kinds of implications for societies that have to be dealt with by people other than scientists. I mean, this is an issue for all of us. What do we want for ourselves as a society? I mean, personally, what I would like is to live healthily till at some point that's unknown to me till I get there, I just drop dead in my sleep. I think that would be a good way to deal with the situation. And I suppose that's the nearest I can describe to what we're trying to achieve. But of course you're right, there are big ethical implications, and I think we have to consider them collectively. That's not a matter just for scientists. But I would say that throughout the history of medical research and medicine, we've regarded it as an ethical imperative to try and help people to improve their health. And most people who have got health problems now are in the advanced stages of life, and I don't regard the imperative to help them as any less than it was when the burden of ill health fell predominantly on the young.
Thanks for that question. Question here, and then I'll take one in the middle, and then there's somebody here wanting to ask a question if we get a microphone down.
Michael Ackland, I'm wondering what the current learnings may be from premature ageing conditions such as progeria.
That's a fascinating question. So there are various human genetic syndromes that result in what looks like very rapid aging. Some of them more severe than others. So some of them have an onset even after 2 years, and you can have teenagers who look very aged with low fat deposits, hair loss, and so on. Some of them are slower than that. And the question is this really accelerated aging? Can we use what we know about aging to help those patients? And conversely, can we look at them to understand what happens during normal? It's gone backwards and forwards, that one. Is what we're seeing here a novel pathology that's got nothing to do with the normal aging process? Or is this really an acceleration of everything? And certainly all of them have some of the features of aging, but not all of them. And I think the most interesting one is something called Hutchinson-Gilford progeria, which involves changes in the membrane, if you like, that surrounds the nucleus of the cell. And people said for ages that's just something odd that happens in this syndrome. But increasingly it's looking as though it's a feature of normal aging. The nuclear envelope actually becomes leaky just during the normal aging process. So there may be actually commonalities between the two.
You can certainly feel it when you get up in the morning, I can tell you. Yes.
I'm from Kawartha College, and I just wanted to ask, how do you weigh up the benefits of a longer lifespan with the problems of overpopulation?
How do you live with yourself, Professor Partridge?
Well, fortunately, I'm not a policymaker or a demographer, so in some senses I don't concern myself with that problem. But as I understand it, at the moment, in most populations, the major determinants of population size are actually the birth rate, not the age at death. So if you look at population number changes in different parts of the world, in populations that are increasing, they're being driven almost entirely by increases in birth rate, not by this increase in lifespan that's been going on. So I think it's a relatively minor player, actually, in the problem that you're talking about.
Which brings me to, just before we take some more calls, too much radio. You mentioned the extrinsic factors in your talk, and you talked about medications, and there was a question about simvastatin. But what about public health type extrinsic factors and the influence on this system that you've been looking at? Because people are living longer, they're living longer and healthier, and if your research interests, your area is right, it must be having an influence on this system, if it's working in humans. So not smoking, taking exercise, eating better.
I would say that on the whole, those kinds of factors that have removed problems from very different directions. So the main increase in human life expectancy, not the one that's happening now, of course, has actually come from dealing with infectious diseases. So immunization and antibiotics, that would produce the huge intermediate increase in life expectancy. I think most people don't understand exactly what's producing the increase in survival in the older age groups at the moment. It's sometimes direct medical care, but it's not clear exactly what's feeding in there. And I don't think we know if it's anything to do with the system.
So you haven't examined mice or in a running wheel and IGF or anything? Yes, question at the back, and then we'll come to a question here.
Thank you very much for the talk. I just want to know why the experimental units die in the experiments. Do they die because of the cancer, or like, is there any other disease that we are not aware of, or something like that?
It depends on the animal that you're looking at. So, of course, humans have been very intensively studied for a very long time, looking at causes of death and the kinds of pathologies that produce it for medical reasons. And often by analogy, we know almost the same amount about mice. Until very recently, nobody's been terribly interested in why a fly dies. So it's very sad, I know, but there's not a great deal of aging-related pathology on the invertebrates. So I think it's fair to say we don't really usually know why they die. It's very clear that worms get problems with their musculature, and it seems more that flies get problems with their nervous system, but we don't know enough about it. In mice, they mainly die of cancer. That's the predominant cause of death. They don't suffer so much from cardiovascular disease. They get a lot of kidney disease and also quite a bit of problems with the nervous system. Those are the main causes of death, and they're the main ones that are shifted, for instance, by dietary restriction. Dietary restriction has a dramatic effect on the incidence of cancer. It really cuts it down.
Thank you very much. And a question here?
In Australia, the biggest issue politically is carbon. And I'm 70. Will old age get me before global warming?
So,
yes.
Are you living at Brighton? Then you might have...
Yes, I hope the young people in the room are listening to this when they're deciding what research area to go into. I mean, what is the real important problem of the day that might be solved by science? And I havered between the two, and I agree with you. My other life actually is work on climate change in fruit flies, because they're rather a good indicator species in terms of stress tolerance and geographic range. And it was my other big research interest until recently, for exactly the reason you give. It's the huge policy issue that we face.
Two questions at the back, and we might have to wind up shortly. Yes, here.
Hi, I'm Rosie from Kerry Baptist Grammar. I was just wondering, if such a pill was to become created and legalised, would you expect it to be provided by the government, or would you expect people to buy it? And if so, do you think this could create a divide between those who have access to such a pill, or I don't know, could you just comment on this kind of idea? Thank you.
Yeah, standard question about access to medical health care. Obviously, the governments would have an interest in providing such a pill if the health economics worked out from their point of view. I mean, if one was just looking at it as a brutal financial issue, if it saved them more than it cost them, then it would be provided free. Otherwise, the normal tensions would apply. I mean, obviously, my own belief is that everybody should have access to the fruits of medical discoveries.
As long as they get enough superannuation to cover it. Question at the back, yes.
You said earlier in a answer to one of the questions that it was very much about quality of life. I'm wondering if anyone has considered studying the 2 mice that were floating on their backs. Seems to me that they've got it sussed.
Cool Californian laid-back mice.
I think you're so right. You know, I've never had that question before, and I think it's rather an Australian question.
And sorry, one final question at the back there.
Len Harrison, the Walter and Lysol Institute. I want to ask you a question about inflammation, but just as an introduction to the question, can I say to the questioner from RMIT that statins are the most expensive anti-inflammatory drugs known. Okay, they might lower cholesterol, but they're very effective at inhibiting inflammation. So my question, Professor Partridge, is you didn't mention inflammation very much in your talk, but in response to Norman, you did acknowledge that all these conditions are associated with low-grade inflammation. So my question is, do you think that inflammation is simply an epiphenomenon, or do you think that it actually is a direct mediator of some of these processes that lead to the ageing effects, like insulin resistance and atherosclerosis and so on? Because you didn't really touch on that very much in your talk.
Yes, I think that's a really tough question and a very important one. I mean, one of the hallmarks of aging in all these organisms is an increase in inflammatory markers. Even in the invertebrates, the immune peptides go up with aging. And it doesn't seem to be an indication of any sort of chronic infection. It's not that, you know, bacterial or viral infections going up with age. It's just something that happens as a result of aging. And at the moment, nobody understands why it happens or what its functional significance is in the whole aging process. People have done things like feeding animals antibiotics and looking at whether that reduces the response. It doesn't. Looking at the cost of the immune response, if you can somehow tamp it down, does that ameliorate various aspects of the aging process? Not clear. I think it's one of those areas where there's clearly some extremely important bit of information that's going to come out, but it's not there at the moment. The information is really confusing.
Still lots of angles for the Hall Institute. Len, you're okay. Final question from me. When you're lying there, you know, just drifting off to bed and thinking through the future, thinking of the future, and we've solved a lot of problems, what do we die of with aging? If pure aging is what we're left with, what will we die of?
Do you know, I don't think we know, because one of the extraordinary things about the animal models, when we really ameliorate aging, and still the best intervention we've got is dietary restriction, it's often very hard to say what the dietary restricted animal died of. It just died. It was fine one day and it was dead the next.
Got fed up saying,
just said,
take me away from this horrible diet. Please thank Dame Linda Partridge. That was fantastic. Thank you very much indeed. It's been a pleasure.
Thank you.
Memorable. Thank you very much indeed. Could you please join me in thanking the sponsors, NICTA, Melbourne School of Engineering, Monash, RMIT, Swinburne, CSIRO, the Victorian Endowment for Science, Knowledge and Innovation, Victorian Life Sciences, Computation in Centre, Faculty of Medicine, Dentistry and Health Sciences, University of Melbourne, Bionic Vision Australia, Qantas, Ansto, Phillips, Osmond Fitzpatrick, University of Ballarat, Club Melbourne, Melbourne Health, Bio 21, and the ICT for Life Sciences Forum. Without them, this would not have been possible. Please join me in thanking them. Please stay for an insulin-inducing dessert and have a good time. Thank you very much.